Hi all, welcome to free license class. So in this video we are going to analyze questionnaires of previous year question paper, that is from CSIR UGC NET Lifescience examination. So if you liked my video, please give me a thumbs up and also subscribe my channel so that you will get more notification when I upload new series of questionnaire analysis. Let's move. So the first question is, unemployed females with only one X chromosome is a characteristic of individual with option A Cry-Duchat syndrome, B Klinefelter syndrome, C Down syndrome and D Turner syndrome. To solve this question, so in the question it is asked to find the unemployed females. So we have to find the disease which will occur only in females. Okay, so let's analyze each of the disease and conclude our answer. Okay, so the first syndrome is Cry-Duchat syndrome. It's also known as Fabry syndrome. That means it is occurring in the chromosome number 5. So actually it is occurring in chromosome number 5. We can eliminate this option. A second option is Klinefelter syndrome. This is a genetic condition which is occurred in boy. Again we can eliminate this answer, and this boy consists of an extra X chromosome, so he will have XXY. Then the next one is Down syndrome. It is a trisomy or it is a genetic disorder caused when abnormal cell division result in extra genetic material from chromosome number 21. Okay, then moving to the Turner syndrome, a condition that affect only females. Okay, the next one is it result when one X chromosome is present, that is absence of one X chromosome, X0 condition. So from the above options we can find our answer, that is option D, Turner syndrome is the correct answer for this question. Moving into next question, which of the following food crops has recently been genetically engineered to zero in edible vaccine to develop immunity against hepatitis B? Option A banana, option B maize, option C potato and option D tomato. So the crop which develop immunity against hepatitis B. Let's analyze the answers first. What is edible vaccine? Edible vaccines are nothing but transgenic plant and animal based production of or those contain agents that trigger an animal's immune response. In simple, plant or animal made pharmaceuticals are edible vaccines. In 1989, the effort to produce a plant-based vaccine was formulated by Hyatt and co-workers. In 1990, Dr. Aronson introduced the concept of using transgenic plants to produce and deliver subunit vaccines. So this idea of Aronson proved that edible vaccine can annihilate the restrictions in the production of traditional vaccines. In tobacco plant, Streptococcus mutans, a surface antigen, is expressed from hepatitis B by Mason et al. Is the milestone in edible vaccine production. In parallel to the production of edible vaccine in tobacco, they also started the production of hepatitis B and heat labile toxin B in potato and potato plants. So from the last sentence, we can conclude that our answer is the potato. So potato plant is used to make or develop immunity against hepatitis B. So the next question, a most commonly used method of estimating primary productivity of a pond involves a measurement of the amount of option A carbon dioxide utilized, option B autotroph biomass, option C oxygen released and option D organic carbon. So it is asked to find the primary productivity of a pond. how we find the primary productivity or how we measure the primary productivity? Primary productivity is measured from the amount of oxygen consumed by a volume of water in a fixed period of time. So water for which productivity is to be determined is enclosed in sealed white and dark bottles. So from the above statement, we can conclude that option C is the correct answer. Oxygen released is used for measuring the primary productivity in ponds. Okay, let's move into the next question. The area of allowed regions in the Ramchandran map will be leased for option A glycine, option B L-alanine, option C L-proline, and option D alpha-methyl L-valine. So, always student will go for option C, that is L-proline, which is the restricted one in Ramachandran plot, but please look to the option D also, that is alpha-methyl L-valine. Here the backbone of valine is restricted by a methyl group. So the answer for this question is alpha-methyl L-valine. So we can look to the structure of valine first. So here in this structure you can see a hydrogen in the structure of valine. So it is changed by this hydrogen is changed to methyl group. It will make more steric hindrance in the compound, so that it will cause or it will restrict the rotation along the backbone more than the proline, because proline can occur both in cis and trans conformation, but in the case of alpha methyl L-valine, it can only go for a trans conformation. So most restricted one in the Ramchandran plot is alpha methyl L-valine, and most allowed one is glycine, amino acid glycine. Okay, let's move into the next question. Small RNAs with internally complementary sequences that form hairpin-like structure synthesized as a precursor RNAs and cleaved by endonucleases to form short duplexes are called option A snRNA, option B mRNA, option C tRNA, and option D miRNA. So the correct answer is miRNA. Why? Because miRNA is synthesized as a precursor molecule. It consists of a hairpin loop. After that, this hairpin loop is cleaved to form a functional miRNA. Okay, so the correct answer for this question is miRNA. In this diagram, you can clearly see the loop here, and this loop is then, hairpin loop is then cleaved to form a functional miRNA. Okay, let's move into the next question. Polar bears maintain their body temperature because they have more of option A transducin protein, option B uncoupling protein, option C myoglobin protein, and option D F0 F1 ATPase. So how does this polar bear maintain their body temperature? It is because they have a high amount of uncoupling proteins. Okay, let's analyze each of the options. First one, uncoupling proteins. Uncoupling occurs naturally in brown adipose tissues. So this tissue is rich in mitochondria, and the inner mitochondrial membranes of which contain a protein called uncoupling protein or thermogenin. This thermogenin allows H plus ions to flow back into mitochondria without having to enter via the ATP synthase, and so uncouples electron transport and oxidative phosphorylation, generating heat. The importance of this natural phenomenon is that brown adipose tissue is found in sensitive body areas of some newborn animals, including humans, where the heat production provides protection from the cold condition. In thermogenesis by brown adipose tissues, plays a role in maintaining body temperature in hibernating animals. That is the reason the polar bears can maintain their body temperature. Then the next option is about myoglobin protein. It is a globular protein made up of a single polypeptide chain of 153 amino acid residues. It is first two protein that have a 3D structure. That is the first one to analyzed under X-ray crystallography. The next option is F0 F1 ATPase. So this ATP synthase is a large enzyme complex of the inner mitochondrial membrane, and it catalyzes the formation of ATP from ADP and Pi, accompanied by the flow of protons. ATP synthase, that is seen in complex number 5, has two functional domains, F0 and F1, and this F1 a peripheral membrane protein and F0 which is integral to the membrane, and F1 the first factor recognized as essential for oxidative phosphorylation. And the next one is transducin protein. Transducin is a 3-subunit guanine nucleotide binding protein or a G protein which stimulate the coupling of rhodopsin and cGMP phosphodiesterase during the visual impulse. So our answer is uncoupling proteins. Okay, let's move into the next question. Transport of water across aquaporin is regulated by the presence of which of the following sequence of 3 highly conserved amino acid? Option A, alanine, asparagine, proline. Option B, proline, asparagine, alanine. Option C, asparagine, proline, alanine. And option D, proline, alanine, asparagine. So, we know the answer, that is 3 amino acids are asparagine, proline and alanine. We have to find the correct order from the options. So the correct order is option C, asparagine, proline and alanine. Okay. So aquaporins are proteins that is composed of 6 transmembrane alpha helices embedded in the cell membrane with the N and C terminal of the protein present inside the cell. Note that point. Both the N terminal and the C terminal are present inside the cell. Between these 6 helices, 5 loops are present and 2 of them are hydrophobic in nature with an asparagine, proline, alanine. That is denoted as NPA motif pattern. So these NPA motif and the 2 asparagine residues in the aquaporins participate in forming a channel for water molecules that is about 3 Armstrong at the narrowest point, that makes the aqueous channel narrow in the middle and wider at the 2 opening ends. So our answer is asparagine, proline and alanine, which forms a motif known as NPA motif. Okay. Let's move into the next question. Which of the cyclins has essential function in the S phase of the cell cycle? Option A, cyclin A type. Option B, cyclin B type. Option C, cyclin D type. And option D, both B and D types. So, this is one of the important questions in the CSIR exam, because we can see any one of the question from this section. Okay, let's move into the answer. Here you can see in this beautiful diagram each cyclins and CDKs which present in each phase of the cell cycle. So starting from the S phase, here in the S phase you can see that cyclin A and CDK2 are present in the S phase, or starting from the S phase. And the next one is G2 phase. In G2 phase, the same cyclin A and CDK1. CDK is changed. CDK1 is there in the G2 phase. Again in the M phase, it is changing to B type, that is cyclin B with CDK1. After that, in the G1 phase, you can see cyclin D along with CDK4 or 6. Okay. In the intermediate between G1 and S phase, you can see another one also, that is cyclin E and CDK2. So this is the cyclins present in each phase. So please by heart this one, because this is very important. The cyclins and CDKs which present in each of the phase in the cell cycle. So let's move into the next question. So the answer for this question is A type, that is cyclin A is present in the S phase. Next question is, during the generation of an action potential, depolarization is due to option A, potassium efflux, option B, sodium efflux, option C, sodium influx, and option D, potassium influx. Let's analyze a diagram. After that we can answer this question. So here in this beautiful diagram, you can see a first thing from starting from the bottom of the page. You can see hyperpolarized condition which have a charge of minus 90 millivolt, where the channels, potassium as well as sodium channels, are closed. Then moving to the resting membrane potential of minus millivolt, where you can see same condition, a sodium as well as potassium channel is closed. When a stimulus occur, it will change into a depolarized condition. In this condition, first the sodium influx, sodium is coming inside of the cell. So sodium influx is taking place, where the potassium channel is still closed during the depolarized condition. So then moving to the action potential of about plus 30 millivolt, where you can see both the sodium gates are closed but the potassium channels are open. Then after the action potential has made its success, then it will change into a repolarized condition, where you can see again a sodium channel is closed and the potassium channel is open. After that, when it reach again to the resting membrane potential, both the channel closes. This is the overall sum up of the action potential. So the correct answer is sodium influx. Okay, option C, sodium influx, is the correct answer for this question. During depolarization, a first occurring event is sodium influx. Okay, moving into the next question. G proteins linked receptor transmembrane proteins of option A single pass, option B 3 pass, option C 5 pass, and option D 7 pass. So do you know the structure of G proteins? G proteins are transmembrane proteins which have 7 pass or 7 helices. So please look into this diagram. Here you can see clearly the G protein have an N terminal as well as a C terminal, and it spans along the membrane 7 times. Okay, 7 times. So the correct answer is 7 pass. Okay. So let's meet in the next video with question analysis. Until then, bye bye.